Welcome back. We're on to the next part of our discussion of nucleic acid structure and DNA replication, and a little bit later the structure of chromosomes. So we talked about the work of Griffith, the work of Avery, McLeod, and McCarty, and now we're on to the experiments of Hershey and Chase. But let's keep this in the kind of keep the big picture in context here. We're really asking the question: how do we know that DNA is the genetic material? Now, there's three sets of experiments which really led us to the understanding that DNA was the genetic material. At least in more complex organisms. If you look in the viruses, that's not always the case. So we had the work of Griffith, who gave us the transforming principle, Avery, McLeod, McCarty, who showed us the transforming principle was DNA, most likely to be DNA, and now we're going to look at the contributions of Hershey and Chase. And these guys really wrapped everything up for us and gave us a pretty good definitive answer to the question, is DNA the genetic material, which we now know to be the case. So the experiments of Hershey and Chase involved a bacterial virus called a bacteriophage and a bacterium, the common gastrointestinal bacterium, Escherichia coli. So just to give you an idea of the scales of things, an E. coli is about 1 to 2 microns in length. They're kind of rod-shaped bacteria, and they so about a micrometer in diameter. So that's a thousandth of a millimeter. That's a millionth of a meter. So if you think about a meter and you chopped it up into a million little individual equal size segments, that's about the length that an E. coli would be. So here is our E. coli, and let's just outline this fella here. Here is, no, we're going to go the wrong way, so let's go back. So here is our E. coli. Here's the end of the E. coli. This is like the kind of disappearing down at the bottom of the screen there. And up here are some bacteriophage, and they have these like tails, and then they've got these kind of head structures here. And so these are all attached to the surface of this bacterium. And so you can see these bacteriophage, these bacterial viruses, are tiny, tiny, tiny compared to the size of the E. coli. So these viruses infect the bacteria, and once they get into the bacteria, they replicate their genome, they take over the bacterial cell, and they use their own DNA to make lots and lots and lots of new virus particles. So a couple of important things about these bacterial viruses. They're only made of 2 types of macromolecule. They've got these capsids, which are made of proteins. This is like a protein shell. And then inside the capsid, and so my arrow here is actually maybe pointing inside, that is actually where the genetic material is found. So we've got DNA as the genetic material, and then that is wrapped in a capsid protein shell called the capsid. We have the head-like structure, which you just saw on the electron micrograph, and then there's this tail through which the genetic material is going to be injected through the cell wall of the bacteria and into the bacterial cytoplasm. And then there are these little leg kind of structures which are part of the base plate, and they basically are responsible for specifically binding to a certain bacteria, to the E. coli bacteria. So Hershey and Chase had 2 questions they were really asking with their experimental system of bacteriophage and E. coli. And the first question is protein the genetic material? And the other question was, is DNA the genetic material? So at the time, although they had the work and the understanding from Avery, McLeod, and McCarty, and from Griffith, there's still a lot of people who said, well, look, protein is made from 20 different amino acids, so that gives you tons of diversity. That means you can put these things together, these amino acids together, in millions and millions and millions of different combinations, but you've only got 4 nucleotides, so it makes sense if DNA wasn't the genetic material, but the protein was the genetic material because of the diversity you get in the 20 amino acids. So these were their 2 questions. Here's what I want you to do. I want you to write a hypothesis for each question. So pause the video, think about it, and write a hypothesis for each of those 2 questions. Okay, so here's kind of the thing that you should come up with. If I asked you, is protein the genetic material, you'd probably say yes or no, but we often try and phrase these hypotheses positively. So if I said to you, is protein the genetic material, you'd probably say, yeah, protein is the genetic material. Or I'd say to you, is DNA the genetic material? And you'd say, sure, Duncan, DNA is the genetic material. So those would be the 2 hypotheses, tentative, falsifiable answers to the well-framed questions, is DNA the genetic material, is protein the genetic material? So the next step that we could do is we could say, okay, let's write a prediction for each of these hypotheses. So pause the video again and write down a prediction for each of these hypotheses. Okay, now if you can do one, you can do both. So let's just say that we're going to go with the hypothesis that protein is the genetic material. You and I know that's not the case, but let's just go with that as a hypothesis. Protein is the genetic material. That's our hypothesis. So then I would say something like, if protein was the genetic material, then I would expect the protein in the virus to enter the bacteria and be used to make new viruses. That's what the genetic material would do. It would act as a template, a blueprint to direct the bacterial cell to make new viruses. That's what happens in viral infections. They go inside cells, and then the genetic material tells that host cell, hey, you need to make more viruses. So if protein is the genetic material, we'd expect it to go into the bacterium during the infection. Now, if we did the other hypothesis that DNA is a genetic material, we can then equally say, if DNA was a genetic material, then we'd expect to see the DNA inside the bacteria, and that be used as the information-containing molecule to direct the bacterial cell to make new viruses. So the question then comes, how can we tell, how do we determine whether during the infection the protein goes into the bacterium or the DNA goes into the bacterium? One or both of them has to go in. I guess neither could go in. One, both, or neither has to go in. But if we are saying that the protein or the DNA is the genetic material, then one of them has got to go inside the bacterial cell and basically take over the bacterial cell and then have the bacterial cell make new viruses. That would be the role of the genetic material in this system. And so how can we tell experimentally whether the DNA of the virus goes into the cell and acts as genetic material, or whether the protein goes in and acts as genetic material? So let me ask you a series of questions, and you have to think back to our discussion of nucleic acids and proteins, specifically nucleotide structure and amino acid structure. So if you struggle with this, pause the video, open a book, and try and work out the answer. So let's ask a couple of questions. Here we go. Okay, so thinking about that, let's now think about this question. Okay, so based on your answers to those questions is what you should see if you compare the atoms you find in proteins with the atoms you find in DNA. You should find that some proteins, not all proteins, but some proteins, but not DNA. No DNA ever contains sulfur. So there are two amino acids, cysteine and methionine, and they both contain the atom sulfur. You don't believe me? Find a diagram of cysteine and a diagram of methionine, and each of those will contain a sulfur atom. And if you look at the structure of a nucleotide, doesn't matter which one, look at them all if you want to convince yourself, they never contain sulfur. And if we look at the structure of DNA and compare that to proteins, what we see is that DNA will always contain phosphorus, whereas proteins, unless they've had some unusual things happen to them involving cell signaling we talked about, they're not going to contain phosphorus. They contain carbon and hydrogen and oxygen and nitrogen, but DNA contains phosphorus and proteins don't. So what we have now is the ability to discriminate between DNA and protein. DNA contains phosphorus but not sulfur, and proteins contain sulfur but not phosphorus. So we can tell the difference between two molecules if we need to. So here's what Hershey and Chase did based on this knowledge. They had a way of radioactively labeling the proteins in their virus. So they made up a flask of viruses, and there are technical ways you can do that. They got a flask of virus, and all of the proteins in those viruses, in the capsids, in the shells of the viruses, contained radioactive sulfur-containing amino acids. So the isotope of sulfur there is 35 sulfur, so that means it's radioactive. They also did the same thing in a different flask of virus. They made the virus, this time though, radioactively labeled with 32 phosphorus. So they've got two flasks of viruses. One where the proteins are radioactively labeled with 35 sulfurous. The other flask, the DNA is radioactively labeled with 32 phosphorus. Now you can't mix up the labeling here because there's no phosphorus in the proteins and there's no sulfur in the DNA. So you can discriminate between the two. And then they simply ask the question, if we add these viruses to the bacteria, does the 35S radioactivity, which remember is in the protein, go into the bacteria? Or does the 32P radioactive DNA go into the into the cell? So they just said to themselves, does the 35S, the radioactive sulfur, or the 32 phosphorus go into the bacteria? Whichever one goes in has got to be the genetic material. So they set up these two experiments, and they run the experiments in parallel. So we'll go through them side by side, two experiments side by side. So on the left here, Hershey and Chase have taken some E. coli, and they've infected them with their virus with 35S radioactively labeled proteins. So those viruses, those phage, have radioactive proteins. Okay, that's on the left. And on the right, they've got the E. coli in a different experiment, and they've infected those with the virus that have radioactive DNA, and it's radioactive with 32 phosphorus. So they mix these two things together in the separate experiments, and they leave them to incubate for a little bit so the virus can attach to the bacteria and stuff. And then once they've left them for 20 minutes or so, they know the infection has been established, and what they want to then do is remove any virus which is still stuck on the surface so it doesn't kind of confuse our experiment. So they do that using a blender. It's just like the blender you have at home. So they put the bacteria and the virus kind of cocktail, if you like. It's like margarita time with virus and bacteria. They dump that into two separate blenders, and they give it a little bit of a whiz on the blender, and they do it for different times. And so they do it for like a minute, and then they take another set of the experiment and they do it for 2 minutes and 4 minutes and 8 minutes. So they've got this kind of time course, if you like. So what they then do is take their kind of blended bacteria virus mixture, and they put them into separate tubes. And those tubes then go into a machine called a centrifuge. And a centrifuge is just a machine which takes a tube and it spins it really, really fast. So it spins around and spins around and spins around. So anything which is dense sinks to the bottom. And when I say spins around, I'm talking about, 13,000 revolutions per minute, twice as fast as the engine, the pistons in your engine goes up and down. So they spin this mixture of virus and bacteria, these two tubes, they spin them really, really fast. So the really heavy stuff sinks to the bottom. That's basically the bacteria and whatever left the virus and went into the bacteria. So if the DNA went into the bacteria, that's in the bacteria at the bottom of the tube. If the protein went into the bacteria, that's at the bottom of the tube. So what they have is then what's called a pellet. Back up one. Come on, Mr. Pen. There we go. So what happens is in the bottom of these tubes is the bacteria all get pelleted. So they get forced to the bottom of the tube. So we've got the pelleted bacteria. So the genetic material from the virus at this point has gone into the bacteria. So the genetic material from the virus plus the bacteria is down here, or it's down here. We don't know which one yet. And then we have this fluid, and that contains any of the virus which were knocked off of the bacteria during the blending. Okay, so that's the supernatant. That's called the supernatant. It's the fluid which is left over after you've spun all the bacteria out. So what they then did is they took the supernatant, the fluid, and they measured the radioactivity that's present in there. Now think about it like this. If all of the DNA has gone into the bacteria, because it's the genetic material, then there won't be any radioactivity in the supernatant. It will be in the pellet. So that's the kind of thing we've got to think about here. So here's their data. So here's the data, and I'm going to put the data up here, and I'm just going to talk about the axes for a little bit and what the two lines are. Then I'm going to leave you to look at the data for a moment, and then I'm going to ask you a few questions, and we'll talk some of those questions through. Okay, so the y-axis here, this is the y-axis here, and that is the total isotope. Isotope is just the chemical word for radioactivity as far as we're concerned. So the total radioactivity in the supernatant, remember the supernatant is the fluid. It's not the bacteria at the bottom of the tube. And then we've got agitation time in blender. So I just want you to consider the 6-minute time point. We're not going to worry about what the point of doing all these measurements every 1, 2, 3, 4, 5, 6 minutes is. I just want you to look at that one time point there. And so ask yourself, what do these two lines show? And so you can just think about this point here. And that point there is the red line, extracellular 35S. So red is how much 35 sulfur was in the supernatant, and blue is how much 32 phosphorus was in the supernatant. So take a look at the graph for a moment. Pause the video if you have to. I've got a few questions that we'll talk about in a moment. Okay, so let's ask a couple of questions. Does this graph show you, well, let me state it. This graph shows that 32 phosphorus, that would be the DNA, remember, that 32 phosphorus was in the cells. Is that what this graph shows? Okay, so the graph is measuring what is in the supernatant. It's not measuring what's in the cells. It's measuring what's in the supernatant. So this graph shows that there is at 6 minutes, let's say if we just worry about the 6-minute time point here, at 6 minutes, there was a lot more 35 sulfur in the supernatant than there was 32P. So this would be false. This is telling us nothing about what's in the cells. This is showing us all about what is in the supernatant. Now, we could make some deductions. We could say, oh, well, if it's not in the supernatant, then it's got to be in the pellet. So therefore, yes, it's in the cells. But maybe it was volatilized. Maybe it was broken. The DNA was broken down by something and all the nucleotides evaporated. Maybe it stuck to the walls of the tube. So you've got to be very careful in reading the axis. This doesn't tell us anything about what's in the cells. It tells us what's in the supernatant. That's the fluid that's above the cells. Okay, how about this question then? The graph shows that 35S was in the cells. It's the same thing, right? The graph is telling us not about what's in the cells. It's telling us about what was in the supernatant. Okay, so let's take a look at a different question then. The graph shows that there is more 32P outside the cells than 35S outside the cells. So let's just look at this 6-minute time point again and talk this through. There's the 6-minute time point right there. The graph shows there is more 32P outside the cells. So the 32P is the blue line and the 35S is the red line. Now we're talking about outside the cells, so that is the supernatant. So we're okay, our graph is telling us about what's outside the cells. So is there more 32P? Well, here's the blue line, and it's lower than the red line, which is 35S. So this isn't right, this is wrong. It's the other way around. There's more 35S outside the cells in red than there is 32P inside. Remember, supernatant is the stuff that's outside the cells, it's that fluid above them. So let's take a look at this one then. The graph shows that there is more 35S outside the cells than 32P outside the cells. So we're still dealing with the supernatant because we're outside the cells. So we're talking about the supernatant when we ask about things outside the cells. So that's good. So we can draw some conclusions from this graph based on the question that I've asked or the statement I've made here. There is more 35S outside. So 35S, let's just look at the 6-minute time point. Here's the amount of 35S outside. Okay, so that's about 80%. That's 80% of the total 35S, not 80% of everything. That is 80% of the 35S. So if you think about it, there's 100% of 35S, 80% of it is outside the cells. Whereas if we look at this time point here, and let's just call that 30% for argument's sake, 30% of all of the 32P that could be outside the cells is outside the cells. So there is way more 35S outside the cells than 32P inside the cells. So the conclusion from this data is that the 35S was outside the cells, which means that the protein was outside the cells. Now, if the protein was outside the cells, it can't have gone inside the cells to act as a genetic material. Now, if we went back, and I'm sure Hershey and Chase did this, if we went back and we measured the pellets, is what we would have found is there was a lot of 32P inside the cells. And that's because the DNA went inside the cells, and the DNA was labeled with radioactive phosphorus, 32P, and so that went inside the cells. And so the pellet, the cells, would be radioactive with 32P, but there wouldn't be any 35S inside the cells, or there would, but it wouldn't be much. And that's just because of contamination and things getting trapped in the pellet. So is what this led Hershey and Chase to in terms of conclusions was that the DNA entered the bacteria, and it was the DNA entering the bacteria from the virus which caused the bacteria to start making new viruses. And over the following number of years, the mechanism of that got worked out very well. So we'd reached the point that Griffith gave us the transforming principle. Avery, McLeod, and McCarty showed us that the transforming principle was probably DNA. And then we have Hershey and Chase, who kind of wrapped it all together and showed us that, at least in bacteria and viral systems, that DNA was the material that controlled the direction of the synthesis of new organisms. And I hate that word, talking about viruses, biological entities. They really convinced us that DNA was genetic material. So we'll wrap up there. This is a long one. I'm sorry, there'll be another long one, then a couple of shorter ones. So I'll finish up there, and I'll see you next time.